Okay, and whenever you are ready, Brandon.
Great. It is really a pleasure to be here. I'm Brandon Obunu. I'm a member of the Board of Directors of the Genetic Society of America. We have another exciting GSA Awards seminar today featuring the esteemed Mike Lynch. Mike Lynch was awarded the 2022 Thomas Hunt Morgan Medal for his lifetime contributions to the field of genetics, which recognizes the full body of work of an exceptional geneticist, along with a strong history as a mentor. Today, we have the pleasure of hearing Michael speak in more detail about his work. Michael Lynch founded the field of evolutionary genomics, developing the concept of functional diversification by subfunctionalization in duplicate genes and contributions to evolutionary quantitative genetics and conservation genetics. His work has consistently integrated theory with empirical study. He developed the concept of the mutational meltdown that plays a central role in explaining the short persistence of asexual lineages, endangered species, and managed populations. He also developed the concept of subfunctionalization, whereby mutations in regulatory regions preserve both duplicate genes, making possible their functional diversification. Recently, his work conceptualizes the drift barrier, the limit at which random genetic drift offsets otherwise universal natural selection for improvement of DNA replication fidelity. Mike Lynch is director of the Center for Mechanisms of Evolution at Arizona State University, where he works on the 5,000 Daphnia Genomes Project, global patterns of genomic and cellular diversity in ciliates, and especially paramecium, the evolution of replication and transcription error rates, the evolution of transcriptional vocabulary, long-term microbial evolution after regimes differing in population size, mutation rate, and degree of mutational background. Mike has authored or co-authored 3 highly cited books, and I know for me personally, important in my own trajectory: Genetics and Analysis of Quantitative Traits, The Origins of Genome Complexity, my favorite one, and Evolution and Selection of Quantitative Traits. Together, these books provide resources of an unprecedented, comprehensive, and integrative nature to the genetics community. His recent work, The Origins of Cellular Architecture, is a founding opus for this field. In addition to being a past president of the Genetic Society of America, Michael is a member of the U.S. National Academy of Sciences, the American Academy of Arts and Sciences, the Society for the Study of Evolution, the Society for Molecular Biology and Evolution, and the American Genetics Association. He has served on dozens of NIH and NSF study sections. Within the GSA, he has been a strong spokesperson for population genetics and founded the Population Evolutionary and Quantitative Genetics Biennial Meeting. Well, he will accept the award in June of this year, and I hope to meet him there personally. In short, he has been an outstanding contributor to the development of genetics as a discipline and to the GSA in particular. Before we hear from Michael, I have 2 quick housekeeping items. The first is that all GSA online events are covered by a code of conduct. And the link for that is available in the chat. The second is to ask you to put your questions for Michael in the chat, and we'll get to as many as possible in the Q&A session at the end. Welcome, Michael. And without further ado, I will hand the microphone over to you.
Well, thanks very much for that introduction, Brandon. Hopefully everybody can see the slides coming up now. This is quite an honor for someone who has no formal training in genetics or evolution. So full disclosure at the outset here, we have been interested in a wide variety of problems, as Brandon pointed out, in the field of evolutionary genetics and moving into cell biology these years. There's 2 big areas that biologists have obsessed about forever since the founding of the field: diversity and beauty among different phylogenetic lineages. This is just an example in the lower left of a variety of different ciliate species. Almost always the explanation for diversity and beauty is based on adaptation and natural selection arguments. We also have a great deal of interest throughout biology in trying to identify general scaling relationships that transcend species boundaries. This is a famous one here. This is a relationship between the rate of population increase and the size of organisms on a log-log plot. Again, here the explanation for these kind of scaling patterns is almost always based on arguments with respect to natural selection or biophysical constraints. And although natural selection is the most powerful force in the biological world, it's not all powerful. And what selection can and can't do is ultimately determined by what we call the population genetic environment. Now, most of evolutionary biology, evolutionary genetics focuses on this kind of the environment, the external environment down here. We worry a lot about finding mates, getting resources, avoiding predators, avoiding pathogens, and so on. And these are certainly major agents of natural selection. Cell biologists worry about the internal cellular environment, historical contingencies. Evolution is based on the kinds of molecules that were selected to be used over 3 billion years ago. We can't avoid the laws of biophysics. We aren't allowed to break the laws of physics. As we'll see, molecular stochasticity, small numbers of individual molecules in cells can also play an important role. But ultimately, what natural selection can and can't do is dependent on these 3 dimensions of the population, genetic environment, all of which I'll talk about today. So mutation, of course, is the ultimate source of all variation upon which natural selection operates. Recombination reassorts variation within and among chromosomes. And random genetic drift is the noise in the evolutionary process. It dictates the magnitude of selection coefficients on individual mutations that selection can and can't take advantage of. I wanted to point out that Thomas Hunt Morgan, in some sense, he and his descendants laid the foundations for each one of these aspects of the environment that dictates how selection works. Founding the field of population genetics, the descendants here would be people like Herman Muller and Dobzhansky. Cellular genetics, Morgan himself, Sturtevant, and others, and ecological genetics, again, going down the path of Dobzhansky. What will underlie almost everything I talk about today is related to a thing I call the drift area hypothesis, which is a general hypothesis about the limits to what natural selection can and cannot do. So imagine we have some trait that's under relentless positive selection. So selection's always pushing us up to this level of molecular perfection. That's the biophysical limit, which we aren't allowed to break. And then imagine the same selection operating a large number of populations of different sizes, from a small population here over to a large population size here. Random genetic drift is the noise in the evolutionary process. So as we move to larger and larger population sizes, selection becomes more and more efficient, and we get closer to this biophysical limit. But we're always getting to this point where there's a balance between the noise induced by random genetic drift, and in some cases by mutation bias, and the strength of positive selection. So if you had the luxury of studying populations, the same trait under the same selection, the only difference is difference in population sizes. If you could follow a population sampled from different points along this gradient over time, and then assess the mean phenotype or the trait performance, say every 1,000 generations or so, you would ultimately get some distributions color-coded according to the population sizes that look something like this. So as we get to larger and larger population sizes, we're moving from the left here over to the right. The mean performance increases. In general, as we move to the right here, larger population sizes, as we'll see, is correlated with smaller organism sizes. So you'd expect microbes to be over here, and us big clunky vertebrates and land plants to be over here. These ideas can be applied to any number of different problems in evolutionary biology. One of the things we've been interested in for a while, for example, is why certain enzymes appear as monomers in some lineage and dimers in others, not because they've evolved a new function at the interface, but simply because proteins have become more and more adhesive. So you can imagine a gradient in allelic types across here, where we're moving from the left, a pure monomeric type, to a dimer with stronger and stronger interfaces. And U is the rate of mutational origin. of new binding sites, and V would be the rate of loss. There might be a selective advantage to moving to the right, or there could be a selective advantage to moving to the left. But the bottom line is there's going to be some general mutation pressure in one direction, selection pressure in the other direction. And this ultimately dictates the shape of those distributions that I was just showing you. So the two key parameters from a population genetics perspective is the ratio of the upward to downward mutation rate, the degree of mutation bias. And the second thing here, S, is the strength of selection divided by the power of random genetic drift. And that, by moving this up into the numerator, the key parameters S times NE. And everything depends on this one composite parameter, which is a function of mutation, drift, and selection. And the key points that I want to emphasize here, and will keep coming up repeatedly, is you do, in fact, get these distributions. The only thing differing here is perhaps NE is getting larger and larger as you go to the right. But the important point here is that you will get substantial variation in mean phenotypes among populations, even within constant population genetic environments. And then you'll get a gradient in mean phenotypes as you move to populations of different size. And the other point to emphasize here is the most common state is generally not the optimum state. In this particular case, the optimum is way over here, and nobody gets here because of the prevailing power of random genetic drift, and in this case, mutation bias on top of that. So drift barriers have been arguments made, maybe not using the explicit words, but the concept has been used in other areas of biology over the past few years. Maybe one of the first uses of the concept that goes back to W.D. Hamilton's theory for the evolution of senescence. Here, the idea is simply that if you had mutations that operate later and later and only late in life, eventually most of individuals' fitness is expressed early in life. And so late acting genes will be under much less strong selection, and we'll just naturally get the accumulation of deleterious late acting genes. And that will be a natural passive way in which senescence evolves. Protein biologists have been interested in a couple of issues for a long period of time. One is that the folding stability of proteins is on what people call the marginal stability. Generally, proteins are not well protected against misfolding and destabilizing mutations. Usually only one or two amino acid substitutions are required to really destabilize the protein. And also we know now that even though enzymes are very, very good at what they do, they're orders of magnitude below what would be possible under the biophysical limit arguments. I'll be talking today about specific aspects of the drift barrier associated with aspects of genome architecture and the proliferation of error rates and the evolution of error rates across the tree of life. So first, I'll be talking about what we've learned about gene structure and genome evolution over the past few decades, and then I'll be moving into cellular error rates at the level of DNA and messenger RNAs, new area we've been moving into. So just to start by demystifying the concept of random genetic drift, it's simply very similar to the coin flipping problem with an interesting twist, as I'll mention in a second. But imagine when you have a population of a small population here of two alleles, the black ball and the white ball. We start out with a frequency of 50%. Population has a gamete storm, and we pull out 10 new progeny here, and whoops, the frequency is flipped up to 0.6, and we keep doing this repeatedly. And you can see that there's fluctuations for generation to generation in the allele frequency, and eventually we'll lose one of the types or the other. And the important point to realize here is drift isn't just a small population phenomenon. It applies to any population of any size. The only thing that changes is the magnitude of these fluctuations. So here we have a population size of 25, and eventually this particular allele goes to fixation after about 40 or so generations. Now we multiply the population size by 10. The fluctuations are still there, but they're more and more gradual, and multiply the population size by another factor of 10, and they're still there. Effectively, the magnitude of these fluctuations is inversely proportional to the population size. And what's important in evolution is the magnitude of these fluctuations exceeds the deterministic power of natural selection, then random genetic drift will win. The eyes of natural selection will be glazed over by this prevailing power of random genetic drift. So one way to think about this would be, imagine a locus where there's a deleterious allele down here and a beneficial allele here, and they're capable of mutating back and forth to one state versus the other. And then we imagine what the average frequency of that allele would be as a function of absolute population size. At low population size, you'll be at the beneficial allele will have a very low frequency because of prevailing mutation pressure towards the deleterious allele. But as we go to larger and larger population sizes, we'll eventually get to the point where the locus is almost fixed for the beneficial. And you can see there's this pivot point here. That pivot point is the population size that's roughly equal to the inverse of the selection coefficient. That's the point at which the power of selection equals the power of random genetic drift. And of course, if the selection coefficients are smaller, this pivot point occurs earlier and earlier and earlier. So it's all quite simple to think about this if there's the free recombination among different genetic loci, which is the assumption here. And this is a problem we have in biology, that most of our nucleotides, or all of our nucleotides, are linked together on chromosomes. And this causes a significant problem for natural selection. The problem is that now we have mutations arising at linked sites. So we have some beneficials and some deleterious mutations. In this case, you can think of there being 4 different haplotypes, all have 3 beneficials and 2 deleterious. And natural selection, as long as there's no recombination, natural selection will not be able to distinguish these. It will operate on the whole haplotype linkage block, and that makes selection less efficient. As a consequence, this flattens these curves that I previously showed you, making an even stronger gradient and the response of a beneficial mutation to the absolute population size. In effect, owing to the fact that our genes reside together on chromosomes, populations behave in an evolutionary sense as though they're much smaller than their actual absolute population size. So this reduces, again, the efficiency of natural selection. This becomes important because the rate of recombination varies in a very specific manner across the tree of life. Here we're just looking at the situation in eukaryotes, and we're looking at the meiotic recombination rate, essentially the crossing over rate per unit physical distance as a function of genome size. And data here are taken from genetic maps from 100 or so different species, ranging from unicellular species here, that's Saccharomyces cerevisiae, and then we're moving in to larger and larger genomes of land plants and metazoans. And you can see that the recombination rate per physical distance is declining. It's a log-log plot, and as referenced, these dashed lines are slope of -1. So there's an inverse relationship between the rate of recombination per physical distance and genome size. And this is a consequence of one of the major rules that we have in eukaryotic genetics. There's roughly 1 to 2 crossovers per chromosome arm. As genomes get larger and larger and larger, they get larger by expanding the average chromosome length. There's not a typical gradient in more and more chromosomes. So if you double the length of a chromosome, you reduce the amount of recombination per physical distance by 50%. The vertical variation here is due to differences in chromosome number among organisms. So everything's a function of the average chromosome size, and we can predict the average amount of recombination to an accuracy of in excess of 99%, just based on average chromosome size. So all these results suggest that natural selection is doing everything possible to minimize the amount of recombination per chromosome. We apparently need at least 1 chiasma to complete an effective mutation, effective meiosis. Now let's return to the concept of effective population size. I won't go into the details of how we get these numbers, but it comes from measures of polymorphisms in natural populations, and you can tease out from the standing variation the effective population size if you happen to know the mutation rate for your organism, which we now do for a wide variety of organisms, as we'll see in a few minutes. What I've plotted here is another one of these log-log power law relationships. Here's the relationship between the effective population size of a species and the adult dry weight in a species. The slope here is -1/5, so minus 0.2 scaling on a log-log scale. To the right, we have vertebrates. Then we're moving to the left, land plants, invertebrates, unicellular eukaryotes, and prokaryotes. So there's this negative association. The larger the organism, the smaller the effective population size. This becomes important because of this pivot point. The effective population size dictates how efficient natural selection can be. So if you're down here, which historically the human population has been, not today, of course, but the effective population size down here for a lot of vertebrates is around 10 to the 4th. What that means is that natural selection is incapable of eliminating any deleterious mutation with an effect less than the reciprocal of that, so less than 10 to the -4. And beneficial mutations with advantages less than 10 to the -4 can't be taken advantage by natural selection. Now, if we move to the top, this is really the upper limit to the effective population size of any known species, about 1 billion individuals, which of course is much lower than the actual head count in the species. But again, what's depressing effective population size is the fact that genes are linked together on chromosomes. If you happen to live up here, what this is saying that all mutations with absolute effects greater than 10 to the -5 can be utilized by natural selection. So much finer grained evolution is possible in microbes than in big multicellular organisms. The general conclusion here is that for random genetic drift not to play a role in evolution, all mutations would have to have fitness effects either smaller than 10 to the -9 or greater than 10 to the -4, and that's an implausible situation. In fact, we have plenty of data now suggesting what the distribution of fitness effects of mutations are from population genomics. This just happens to be one example from Daphnia work. I don't want to go through all the details, but these are distributions of this ratio of selection to drift, where the pivot point again will be a value of 1. Different flavors of nucleotides in the genome. And you can see that much of these distributions are to the left of this pivot point. So many, many mutations in natural populations have these very, very small effects, which means selection can take advantage of them in some contexts, but not in others. Even not worrying about the population genetics, we can calculate the selective consequence of certain kinds of mutations from first principles. So for example, think about an insertion of a single base pair into a genome. We know from biochemistry that to make 2 nucleotides, to biosynthesize 2 nucleotides, it costs about 100 ATPs. We know that for E. coli size cells, the total lifetime budget is about 10 to the 10th ATPs. So the ratio 10 to the -8 can be thought of as the negative selective consequences of adding a single nucleotide to an E. coli size genome. And if the effective population size is bigger than 10 to the 8th, a microbe like E. coli could actually feel over evolutionary time this single base addition. And I'll follow this up now by thinking about genome architecture and gene structural evolution that we've learned a lot about over the past few decades. So to the left, we're looking at the gradient in gene structure complexity. Prokaryotes are like the Priuses of the genomic world. Nice single open reading frames, often in polycistronic units. So there's only one regulatory region for a series of different genes. There's no intronic DNA, very little transposable element associated DNA, and so on. And then we get into eukaryotic genomes, where our genes tend to be subdivided by large numbers of introns. Not uncommon to have 7 or 8 introns per protein coding gene. Often these intron lengths are much, much bigger than the tiny islands of exonic DNA, complex regulatory regions, and so on. So much more complicated genome architecture, gene structural architecture in eukaryotes. And that means that our genes are much more easy to break mutationally than the genes of prokaryotes. For example, you have to retain certain nucleotide sites for the proper signature for splicing. We have to worry about transcription factor binding sites and so on. There's many more ways to break. These are mutationally hazardous ways to construct your genes. We've also learned a lot about the gradient in genome architecture now that we've sequenced thousands and tens of thousands of genomes. So this just gives you a general idea of how different components of the genome scale with total genome size. And these are just isoclines. So if you were up here, 100% of the genome would be there. And if we're looking at coding exons, all bacteria would be up along this line. About 95% plus of bacterial genomes are coding DNA. And then as you get into eukaryotes, initially, as genome sizes increase, the amount of coding DNA continues to go up, but eventually it levels off. And so that as genomes get larger than about 10 million bases or so in eukaryotes, a further increase in genome size is generally due not to increase in coding DNA, but non-coding DNA. So it's intronic DNA, dead genes sitting around the genome, and all the different flavors of transposable elements and retrotransposons. So you get this massive expansion in genome size. And we've thought about this quite a bit recently as to why you might get this gradient, because these are not components of the genome that are in general doing anything beneficial to the genome, but it's an energetic burden on the cell to carry all this excess DNA. And so we've been giving some thought to how much it costs to add an addition of DNA that may or may not be transcribed and translated to a genome. There's a lot of biochemistry, molecular biology involved here, but if you think about an insertion of a piece of DNA, there's going to be cost at the DNA level, the cost to synthesize the nucleotides. If you're a eukaryote, we've got to wrap those nucleotides around proteins called nucleosomes. There's a cost of helix unwinding or replicating, and lots of other things to worry about. I'm just trying to give you general flavor. Transcription, the piece of DNA is transcribed, we have to worry about the synthesis of the ribonucleotides. Transcripts turn over very rapidly in cells. We have to cap them, we have to splice them, we have to polyadenylate them. All these things cost in terms of ATPs or GTPs. And of course, if things the mRNAs are translated, and then we have to worry about the cost of amino acids, cost of chain elongation, and so on. And we know quite a bit about all of these events and how much they cost from a lot of years and years of work in biochemistry. If you add all these costs together and then divide by the total cost of a cell, as shown in the example before with the E. coli single base insertion, we now have a measure of the selective burden of the genomic embellishment on the organism, essentially a measure of the fractional reduction in the growth rate of an organism. We know now how much it costs to build a cell from a variety of studies and microbial physiology. I won't go through the details, but it basically scales, power law relationship, the exponents pretty close to 1. So in other words, the cost of a cell increases proportionally to the volume of a cell, ranging from bacteria to unicellular eukaryotes to multicellular eukaryotes. So we know how much we know something about the denominator, though, now of that ratio that I just wanted to mention. And now I want to think about the cost, the burden that a gene in, well, really the whole population of genes in an organism has on the cell. And then we can ask the question, are organisms in a use it or lose it situation? So they have to use a gene, otherwise they pay the penalty of the metabolic burden of just carrying the gene, and then it'd be subject to deletion, or can we get this passive accumulation of excess DNA? There's a few organisms for which we have all the information to do the bookkeeping for every gene in the genome. You won't be surprised that one of those is E. coli. And what we're looking at here are the histograms, basically, of the 4,000 genes in E. coli and the cost at the replication, transcription, and translation level. Let's just look at the total cost of all of these. This is telling us that the peak is about a cost of a gene, average gene in E. coli is about 10 to the 5th ATPs. So this is the distribution of the lifetime cost of a gene in a cell, and there's variation due to differences in transcription and translation. Now, the upper line here is nothing more than this bottom line divided by the energy it takes to build a whole E. coli cell. So that's the denominator from the previous slide. You can think about that as the selective disadvantage of carrying this gene if it's not doing anything for you, if you have no ecological advantages of it. And then finally, these lines here, the -9 here, that is the drift barrier. We'll assume that regenerous in, say, E. coli has an effective size of about 10 to the 9th. We can add an order of magnitude or so leeway here. But the important point is everything is way far to the right of the drift barrier. And that means that E. coli or bacteria in general can feel the insertion, as I already pointed out. As few as 1 or 2 base pairs, natural selection would be capable of eradicating that based on the energetic consequences. So this provides a nice, simple, finely mechanistic explanation for why microbial genomes are so streamlined. Now let's extend this idea to observations from other organisms here. Not surprisingly, 2 of these organisms are yeast and Arabidopsis, for which we have lots of information. 3 things happen here. The numbers move to the right as we go into larger and larger organisms. That's because genes are more complex and, for reasons I just mentioned, are more expensive to run. On the other hand, the upper axis, the numbers go down, and that's because cell size has become larger and larger and larger. The denominator has become larger, so the selective consequences of adding a DNA embellishment go down. And then finally, the drift barrier moves to the right. So what we're seeing as we move from bacterial size cells into multicellular species, everything moves to the left. And what this means is in multicellular eukaryotes, it's possible to insert things as large as dozens of kb. It's an energetic burden on the cell, but relative to the power of natural selection, it really can't be felt by natural selection. So this provides a nice natural explanation for the expansion of genome complexity passively due to the reduced power of random genetic drift. I'm going to end my talk. Hopefully I can go for a few more minutes here and talk about what we've learned about mutation rates at the DNA level and the RNA level. Let's start by thinking about how selection might operate on the mutation rate in an organism. We have an asexual population here. Mutator aligned arises. Most mutations are deleterious. So this mutator strain here has an increased rate of input of deleterious mutations. Each has a selective consequence of S, so that's the rate of output. And so this mutator lineage will pick up an excess burden of numbers of deleterious mutations. It's nothing more than the ratio, the input to the output. We want to know how bad this mutator strain and its descendants are feeling. So I have to multiply the excess number by the effect S. The S cancels out. And so the total consequences of mutator and anti-mutator turns out to be independent of the individual effects. It's simply a function of the change in the mutation rate itself. And this becomes important because mutation rates have evolved at very, very low levels. So if you want to think about the selective advantage of an anti-mutator, there's very little room for improvement that would be simply equal to the decrease in the genome-wide mutation rate. And of course, you can't go below 0. And if you're already very, very close to 0, this provides a simple argument for why there ought to be a drift barrier operating on the mutation rate. And I won't go through the theory, but the bottom line is that you can start with different mutation rates in an ancestral population. Depending on the population size, you'll gradually evolve, no matter where you start, into this drift barrier area. And there's going to be drift around the drift barrier as anti-mutators and mutators sporadically crop up. What we want to think about now is how does the theory fit with the data? And we've been working on this kind of problem for years with a wide variety of organisms using what we call mutation accumulation experiments. We start with a single individual, clone it up. These are paramecium cells, and take them through single cell bottlenecks for up to several thousand generations. They all started out identical. At the end of the experiment, we sequenced the complete genomes and were able to estimate the number of mutations arising per unit time in each cell lineage. We've done this now with a wide variety. This is an incomplete table now. We've done about 40 species of bacteria, a whole bunch of different eukaryotic species, multicellular and eukaryotic. We're trying to get an idea of how the mutation rate evolves across the entire tree of life. At the end of these experiments, as I said, you sequence the genomes, you count up all the mutations. These are 50 concentric circles for Bacillus subtilis, the soil bacterium, which ends up having a mutation rate about 3 × 10⁻10. Here's another bacterium, Mesoplasma florum, isolated from the petal of a lemon tree. That's all we know about its biology, except for the fact we now know its mutation rate. It has a mutation rate about 10⁻8, way beyond what you see in Bacillus subtilis. To give you an idea, 10⁻8 is the human mutation rate per human generation per nucleotide site. Many years ago, 30 years ago, Jan Drake suggested to us there's an inverse relationship between the base substitution mutation rate and genome size. Here, all his analyses were based on what are called reporter constructs. So this is before our DNA sequencing days, and he suggests there's this inverse relationship such that every newborn microbe would have a probability of 0.003 of obtaining 1 new mutation. And now we can, and Jan had this great line in the abstract of his paper, because this rate is uniform in such diverse organisms, it's likely to be determined by deep, general, mysterious forces, which we believe today are nothing more than the basic principles of population genetics. We can now revisit these kinds of results with measures of genome-wide mutation rate in a wide variety of organisms. Here we're looking at unicellular organisms, bacteria, and unicellular eukaryotes. Everything looks good at this point, with the exception of perhaps 1 point, Chlamydomonas, until you add to the picture multicellular species, and they're not running even in the right direction. So what's going on here? Jake's rule certainly doesn't apply across the tree of life. And we never have really felt that there's a direct mechanistic link between genome size and the mutation rate. What we think, as I've said, is that there's a relationship between effective population size and the mutation rate. And the effective population size I've just shown you is related to genome size. So now if we change the x-axis here to effective population size, everything's running in the same direction, at least. Although we do have 3 different clouds of points, and you can see that unicellular eukaryotes actually evolved to have lower mutation rates than bacteria. That also makes sense in the context of the drift area hypothesis, because unicellular eukaryotes are larger mutational targets. They have larger numbers of larger genes, and that imposes stronger selection for reduced mutation rates. So now, if we modify the x-axis by thinking about the total genome-wide mutation rate, so we're multiplying mutation rate per base times the total number of bases under functional constraints in the genome, if you like, the number of deleterious mutations per generation per genome. Now everything is unified. These dashed lines indicate the slope of -1. There's reasons why these values over here might be undercounted, because we're only looking at the proteomic DNA. But this looks to be now the first general explanation for the evolution of a complex trait across the entire tree of life. Of course, there's not very many traits that you can measure in every organism across the tree of life, but everybody does have to replicate. So this is one of them. Another one that we've gotten into recently is the level of transcription errors. When we want to express a gene, of course, we have to make a messenger RNA, and that has to go on to be translated. Here we've had to do something a bit different. Here we have to isolate individual messenger RNA molecules and sequence the individual molecules. We can't just grow up a jug of DNA and sequence the DNA and count fixed mutations. Here we have to worry about every single event. And here we've developed a kind of a rolling circle way of reverse transcription. So we'll circularize the RNAs, we'll go around multiple times, and then sequence the DNA. And the problem here is DNA sequencing has a very, very high error rate. So we have to factor out all the error rates due to sequencing, and we look in the multiples here for errors that are found in every single interrogation of molecule, and those are true transcription errors. When you do this work, you get enormous numbers of errors to count. So you can, happens to be an experiment with yeast, the distribution of transcription errors over the chromosomes. You can look up and down a chromosome and get the geography of transcription errors along the chromosome. And you can even go to with individual genes and find individual transcription error hotspots in genes. So here's a start code on a hotspot, and here's a non-synonymous site, probably because of the neighboring nucleotide context causes them to have higher error rates than others. The bottom line from this work is that what you might expect from those high error rates is that transcription error rates are on the order of 1,000 to 100,000-fold higher than the DNA replication error rates we've measured in the same organism. As a consequence, about 1%, or perhaps more in some cases, of transcripts in cells have errors. Every ribosome has probably got at least one error in it because it contains on the order of 100 proteins or so. What's remarkable about the transcription error rate, in addition to its very, very low level, is there's very little variation among different phylogenetic lineages. Remember, for the replication error rate, there's about a 1,000-fold range of variation. For the transcript error rate, there's only about a 5-fold range of variation in the error rate across the organisms that we've studied so far, and that ranges up to multicellular species from bacteria. So we're trying to figure out how to explain this in a theoretical context. And you can extend the theory for the replication error rate to the transcription error rate. So remember, I said that the selective disadvantage of a mutator allele is proportional to the number of sites in the genome that matter. That's the genome size. The degree to which new mutations linger around, that's the longevity of a mutant allele before it's eliminated from the population, times the selective disadvantage of each of the mutations that the mutator is producing. And we can think about this in the same context of the transcription error rate. Up here, T can be quite large because this is roughly equal to 1 over the selective disadvantage, the persistence time of a new deleterious mutation. But for transcripts, those transcripts only last a few minutes inside the cell. So this number is orders of magnitude lower than this number. The transcriptome is now what we worry about. The part of the genome that's transcribed, that must be smaller than the genome size. The selective disadvantage of a mutation has to be the same. It's manifest at the protein level just as it is here. But now we have this additional problem here. There are multiple transcripts inside cells. So that's the number. We have to multiply the number of transcripts inside a cell. Up here, we'd be multiplying by 1 for a haploid genome. There's just 1 gene per cell. And then there's this thing we call the dilution effect. If a mutation arises in 1 transcript and there's 10 others, is it felt at all? Is it additive? Is it dominant? Or is it recessive? And we can start to think about how this relates to the so-called cellular environment, because we know how the number of mRNA molecules scales with cell size. Larger cells have more transcripts cycling at any one point in time. So that means there can be more cellular errors inside the cell in large cells and small cells, all other things being equal. So we think about this dilution effect as being 1 over the number of transcripts to the X power. And I'll give an example of this in just a second. The total copy number effect is the number of transcripts times the dilution effect. So if, for example, transcript errors were additive, then the dilution effect would just be 1 over the number of transcripts. You'd multiply it by the number of transcripts, the n goes away and it's equal to 1. So if things were additive at the transcriptional level, it wouldn't matter how many copies. On the other hand, if the effects were dominant, so all you had to have is 1 error, it didn't matter how many there were, then the copy number effect is n, and it's going to be proportional according to this power law relation to cell volume. So I'll finish with this one final slide here, summarizing now all of our data. There's a bit more of a cohesive pattern. We're plotting the transcript error rate in all these different organisms as a function of this composite parameter here, which looks a bit messy algebraically, but in words, the x-axis is just the target size for errors in these different orders. It's the number of protein coding genes times the average transcript, just as target size. The effective population size, the larger that is, we expect the error rate to go down, just as we expect it to go down as the target size increases. And then there's this copy number effect that I just mentioned. The best fit occurs when x equals 0 here in that copy number effect. So that implies that the best fit occurs if transcription errors are operating in a completely dominant fashion. We don't fully know what's going on here yet. We're still trying to explain everything, but this is our working hypothesis. So just to give you an example, to get a better biological field for the placement of these points, Mesoplasma florum, our friend with the high mutation rate before, has the highest transcript error rate. It has a very small proteome size, small mutational target for transcripts, has a very small effective population size, and a small cell size, small number of transcripts per cell. So everything's working in its disfavor in terms of selection for the transcription error rate. Over here, we have two paramecium species. These have very large proteome sizes, 40,000 protein coding genes, twice as much as in the human genome, largest known effective population size of any organism, and large cell size. So at least things are starting to make a little sense in terms of the population, genetic and cellular environments in these different organisms. A lot more work still needs to be done here. So what I'm going to do now is just simply end. The general message that I've been trying to convey here is what occurs in evolution is dictated not just by what natural selection can do, but by what it can't do. Natural selection's relentless search for perfection is limited by the granularity of mutational effects that's dictated by the effective population size. It's defined by patterns of mutational bias as well. At all levels of biological organization, given what we know about the magnitude of selective effects of new mutations, and given what we know about the power of random genetic drift, we expect for all traits to see mean phenotypes scale with the effective population size across the tree of life. We expect there to be gradients in mean phenotypes, even if the trait is under identical selection pressures in all different lineages. And I've tried to demonstrate the potential validity of this hypothesis with a few examples: the passive expansion of genome size and gene architectural complexity as we move to bacteria with very, very high effective population sizes to multicellular eukaryotes with very, very low effective population sizes. We've seen the mutation rate variation across the tree of life is reflected in differences in the power of random genetic drift. But if we want to understand the variation in the transcript error rate, it appears that we need to really be thinking about the cellular environment as well. And then finally, I haven't been able to go into this today because of the time limitations, but we have quite a bit of data now showing that there's a reduction in performance of individual enzyme proteins in organisms with reduced effective population sizes and very definite declines in maximum growth rate. So a wide variety of traits from various cellular levels of biological organization seem to support this hypothesis. And I'll just end by thanking members of my lab over the past few decades. These are a couple of photographs from my lab in pre-COVID days. Hopefully in a year or so, maybe a few months, we can start to take another lab photograph like this. But a great number of colleagues in my lab have gone on to start their own labs, and in many cases have contributed to the thought processes here. We've been very lucky to have support to do the experimental work that I talked about from the NSF, from the Department of Army and NIH, and as well from the Simons and Moore Foundations. And I think that's it. Thank you very much.
Great, thank you. Thank you for that, that thrilling seminar. Congratulations again on the award. Michael. I think right in line with the way you were introduced, just a stirring panorama of just compelling and provocative topics and questions. I will take questions. Those interested can oh, okay, there's some questions already in the Q&A. You can drop them in the chat or the raise hand feature for people who kind of would prefer to kind of verbalize those. I see a question from David Ram. Hey, David, long time. Oh, great. Yeah.
Hey, Brandon, how are you? Hey, Michael, wonderful talk.
Hi, David.
Great to see everyone. Yeah, that was fabulous. Really enjoyed it. I guess my question as a pan-selectionist is, can we invert your argument to say, you know, if the pervasive effects of drift are, and the limits of selection are so clear from those affected population size, can we take the phenotypic differences we see among organisms, even within species, even within humans, to be able to estimate how effective selection has been in generating those phenotypic differences? You know, what I'm trying to do is sort of invert your limits of selection argument and then take a phenotypic approach and say, how strong has selection been in the history of humans or the history of fruit fly wings, or all these really cool phenotypes that are obviously different between individuals? Is that tractable from what you've described?
Well, in some sense, that's what we're trying to do. I think it's easier to think about in the context of an error rate in a cell if we think that the vast majority of errors are deleterious, because then we think of the same direction of selection operating across all lineages. It becomes more difficult when we think about a trait like, you know, wing size or something, because then it's likely that there's a different optimum. In other words, the fitness function isn't going to be the same. So that's really the main reason we've been trying to focus down on the molecular level right now. Many things inside cells, you know, like the Krebs cycle's been around forever, so we can think about certain enzymes having been exposed to the same selection pressures over evolutionary time. But it's a great question. It's certainly something we'd like to think about quite a bit more if you can come up with some good target traits. Growth rate is one that we're working on very hard now. And we're trying to figure out the maximum growth rate in the entire life history of any organism. And there the idea is that selection should be not throughout the life history, but in general, it should be selecting for higher assimilation rates and growth rate efficiency. So that is one we're working on right now. And that might come close to what you're asking about. And there's a real negative gradient. And as you get to organisms of larger cell size, the maximum possible growth rate goes down at a power of about -0.2, which is the same as this power in the NE body size relationship. It may just be completely coincidence, but growth rate is one trait that applies across the tree of life. So that's why we've gotten interested in that one.
Yeah, good. I mean, obviously, yeah, you can't use leaf size in one organism and wing size is another. Growth rate is a standard trait. So yeah, good answer. Thank you.
Yeah.
Thank you for that, David. I see a hand raised from Patrick O'Farrell.
Sorry, I have to unmute myself. Michael, thank you. That was really beautiful and nice. And I just had a question about, so linkage was a pretty big factor in the effects that you described. And then superimposed on linkage, it seems like the clustering of functions could really make a big difference. And so I wonder what your thoughts are about why prokaryotes have operons and we don't. And there's some cases of linked groups of common functions in metazoans, larger metazoans. But for the most part, related genes seem to be all over the place. how do you think selection is operating on this factor, this linkage of common functions?
Well, I guess in general, the way I view things is in prokaryotes is it's as good as it can get because natural selection is so efficient. So in general, the idea would be that it's advantageous to have operons. The advantage might not be that great, great enough for bacteria to hold on to them, but not quite good enough for eukaryotes to hold on to operons. There are, as you I think alluded to, there are some eukaryotes, C. elegans being one of them, that do have some genes that are constructed as operons. So I guess that's about all I could really say on that. It could be, you've got to also think about recombination rates being higher, perhaps, in eukaryotes and breaking up operons. And so you've got to think about that bias just due to recombinational breakup. And if that's higher than the rate, higher than the advantage of an operon structure, then recombinational breakup's going to win. You're muted. Hopefully you haven't been talking too long.
No, sorry. What about the impact of movement of genes and perhaps clusters of genes from organism to organism? So in prokaryotes, you've got transduction effects and other things, and you've got functional units that, if they move together, can drive an evolution that's at a little bit separate from the organism itself. And so we see this effect where you see islands of pathogenesis and pathogenic genes on episomal elements, etc. Is that going to play into this?
Well, I think, as fair to say, there's more and more appreciation for horizontal gene transfer in eukaryotes than there used to be, but it's not a pervasive thing.
Right.
But in principle, I mean, it would be an evolutionary force like anything else, and that's basically how evolution works. You have these competing forces, and if one force becomes greater, you saw it in everything I said. So this parameter NES, that's the ratio of selection to drift, and so on. So everything's sort of a function of the ratio of the power of different forces. So in certain contexts, if the rate of horizontal transfer were great enough, then it could offset some other opposing evolutionary force.
Right. Seeing no other questions, thank you for those excellent questions, and similarly excellent answers. Seeing no other questions at the time, and looking at the 2 o'clock time, we've now we'll conclude our session. And I want to thank you again, Michael, for a stirring seminar, and of course, congratulations again on the great award. I encourage everyone to keep informed on all of the events going on, and a lot of the information you can find in the chat. Thank you all for your time, and have a great rest of the week and weekend ahead.
Yeah, thank you, and thank you to everyone who attended.
I just want to reiterate, thank you, Mike, for joining us today, for giving this amazing speech or seminar. And congratulations on your award. And Brandon, again, thank you so much for joining us today. We really appreciate you taking the time to host this call for us.
My honor.
Yeah, thanks, Brandon.
My honor.